Good evening, everyone, and lovely to see you once again for one of our Oxford University Museum of Natural History online public lectures. Thank you for joining us. Good morning, good afternoon, or good evening, depending where you are in the world. I can see we've got people in from all around the world. My name's Jack Matthews. I work at the Oxford University Museum of Natural History. Firstly, a few bits of guidance about how we use the webinar jam system. If you have any connection issues at any point, please do use the reconnect button at the top of the page, and that should sort out any connection issues you have with the server. Also, you can see on the right-hand side there is a chat function, and people are busy introducing themselves and telling us where they're from in the world. I've already seen we've got people in from India and the USA, Canada, up north in the Yukon, and even someone on their way to Walcott Quarry, which I'm sure our very special speaker will be talking about in a moment. If you have any questions for our speaker during tonight's lecture, do type them into the chat. And on the right-hand side, you should be able to see a little circle, which you can click after you've sent your message onto the chat, and that marks it as a question. And then at the end, I will try and ask as many of your questions as I can to the speaker. Final reminder that our lectures are recorded and placed on the YouTube channel for the Oxford University Museum of Natural History. Give us a few days to get that uploaded, but if you've missed it and you want to pass it on to a friend, please do think about having a look at the past record of the lectures and also sharing this one with someone you think might be interested. And at that point, I'm very pleased to welcome Dr. Derrick Briggs, Professor Derrick Briggs, if he would like to turn his camera and microphone on. And I will get his slides up. Derrick, I should say, started his career in paleontology and geology at Trinity College, Dublin, before moving to the University of Cambridge. Since then, he's been at the University of London and then the University of Bristol before where he is now. He's now the Evelyn Hutchinson Professor of Geology and Geophysics at Yale University, and also curator at the Yale Peabody Museum of Natural History. Derrick has an interest in the preservation and evolutionary significance of exceptionally preserved fossil biotas, and that's, of course, very relevant to our first animals topic. Just to let you know, I think Derrick is having a connection issue at the moment, but we're sorting that out. Derrick is just reconnecting. Derrick's work also involves a range of approaches, from experimental work on the factors controlling decay and fossilization through studies of early diagenetic mineralization and organic preservation. And I'm sure he's going to be explaining some of these terms a bit more in his talk. And he uses a lot of field work on a range of extraordinary fossil occurrences and looks at the ecology and the phylogenetic significance, so where these organisms fit into the tree of life. And at that point, hopefully, Derrick, if you could turn your camera and microphone on, and hopefully we'll be able to see you. Fingers crossed. Derrick, can you hear us? Just while Derrick, we're unable to get Derrick's camera and microphone up, I will just say you should be able to see a link in the chat on the right-hand side for our next speaker. That's Professor Gabriella Mangano. She's from the University of Saskatchewan, further north than where Derrick is, in Canada, a global expert on ichnology, the study of trace fossils. And Gabriella is going to be telling us the story of the first animals told through trace fossils. These are fossils that record evidence of burrows and trails and footprints. So if you were interested in Dr. Shuhai Zhao's talk 2 weeks ago, you will definitely find Gabriella Mangano's talk very interesting. Derrick, can you hear us? Apologies, everyone. We seem to be having some connection issues with the University of Yale at the moment, but we're busy sorting those out. While we are waiting, we will also remind you of the upcoming talks. So we have on September the 9th, as I was just saying, Professor Gabriela Mangano of the University of Saskatchewan. She's going to be focusing her talk on ichnology, the study of trace fossils. And we've got Derrick in the room. Wonderful, wonderful. How are you, Derrick?
I'm fine. We seem to have a little glitch with internet, but hopefully that's going to behave itself now.
Super. We can hear you wonderfully. I'm just going to call up your slides. I've been singing your praises and listing your very illustrious CV already. So we've done the introduction, and we are so pleased to have you joining us again because you've been a great supporter of the museum and gave a similar talk in person. And now we're very pleased to be able to share it with the world in this wonderful virtual format. So at that point, I will disappear, and please, Derrick, take it away.
Thank you, Jack. Well, I do hope we don't have any issues with connectivity. It's a pleasure, as ever, to be associated with an event at the Oxford University Museum of Natural History, one of my favorite museums and somewhere I've spent time in over the years. As Jack will be well aware, the Oxford University Museum was founded in 1860, and I am working at a rather different museum, the Yale Peabody Museum of Natural History. And it's called the Peabody Museum because it was founded with a gift from George Peabody, a philanthropist, in 1866, although it took another 10 years to build the first museum. And the present Peabody Museum was built in 1925. So we can't claim the same historical longevity as the Oxford University Museum. The first, let's call him director, the first curator of paleontology at the Peabody was this man, Othniel Charles Marsh. This is a photograph of Marsh with a magnificent beard. Some of my colleagues are growing similar beards during this COVID lockdown. Marsh at this stage was about 50, and he was the beneficiary of George Peabody's gift because George was his uncle. So there were advantages there. The Peabody, like the Oxford University Museum, is famed for its dinosaurs. And indeed, this Brontosaurus, the big specimen in the middle, was named, as were many other famous North American dinosaurs, by Marsh himself. Now, the Great Hall looks nothing like this right now. Indeed, it's been completely stripped bare because we're in the process of a major renovation of the museum. So I certainly encourage you, if you have the chance, to go to the Oxford University Museum once it's open again. The Peabody, COVID notwithstanding, won't open again till probably 2023, when our renovation will be completed. And we're being extremely careful to look after this very famous Zallinger mural, which was painted in the 1940s, the so-called Age of Reptiles, which is very familiar to many of us as a very early reconstruction of dinosaurs. That's actually painted on the wall of the great dinosaur hall in the Peabody Museum. So it's been very carefully covered and conserved during this process. Anyway, what about exceptionally preserved fossils? This whole concept of soft tissue preservation goes back a long way. Here's a fine quotation, which of course is very out of date now. No organism wholly soft can be preserved. And I wonder how many of you know where that came from. Yes, indeed, it's from The Origin of Species, published, of course, in 1859 by Darwin. And indeed, Darwin was puzzled by the very rapid appearance of animals at the Cambrian, beginning of the Cambrian, which he would have recognized as some kind of explosion. But of course, he was concerned by the fact that there was no trace of fossils in older rocks. Fortunately, now we know a great deal more than he did, and we can begin to resolve that kind of conundrum. So what is exceptional preservation? Well, let me just show you a few examples. It ranges from these extraordinary Ediacaran fossils, late Proterozoic fossils, which are at Mistaken Point in Newfoundland, and which are, of course, in the field. So if you look, you can see that I'm wearing some blue booties here. Here you have them down here. And those booties are insisted upon by the Mistaken Point folks so that we don't damage the fossils that you inevitably walk on when you go on these magnificent bedding plains. Here's a very different kind of exceptional preservation. This is a fossil horseshoe crab that is very obviously so. It comes from a Jurassic locality in Germany, not perhaps the classic Solnhofen, but at a place called Nösplingen. And here on the right-hand side, we can see individual bacteria, spirally coiled microbes, which are preserved here in calcium phosphate, in apatite. And indeed, this kind of preservation is extremely rare because, as you can imagine, bacteria being essentially organic don't have the kinds of features that would lend themselves to ready preservation. Here's a different kind of exceptional preservation. This is a Cretaceous feather from the Crato Formation in Brazil. And here you can see the banding on the feather itself is preserved, black and indeed lighter colored lines. And we can see the way they bleed one into the other, just as modern feathers do. And this is because the melanin encapsulated in melanosomes within the feather is preserved, has a very high preservation potential, even though it's an essentially organic molecule. Here's a very different kind of preservation, rather like those bacteria in the horseshoe crab from Nösplingen. Now we have a specimen which is once again mineralized by very early diagenetic processes, but in this case, a trilobite from the famous Hunsrück slate, again in Germany. And you can see the limbs at the rear of the specimen, limbs that, of course, in the vast majority of trilobites are not preserved because they decay away during the burial and fossilization of the animal. And then finally, as an example, this is, as indeed is probably obvious, a mosquito. This mosquito is from the Eocene of Montana, so about 50 million years old. And what's extraordinary about this is it's demonstrably female, and indeed it preserves the remnants of a blood meal. And work by scientists has shown that this blood meal preserves some traces of the original heme molecule, but also a higher concentration of iron, as you would indeed expect in hemoglobin in blood, than the iron content of the rest of the specimen, or indeed of the sediment that surrounds it. So these examples show you that we shouldn't in any sense underestimate the fossil record. It preserves extraordinary information about life on the planet. So we can look at decay, which of course is the enemy, if you like, of exceptional preservation, in various different ways. We can look at it with fossil examples, and I'm going to show you a series from another Eocene locality, in this case, part of the Green River Formation. And this is a fish called Cocorolites. And you can see here's a very elegantly preserved, essentially complete specimen. And if we watch it degrade with a succession of course, different specimens, we can see that the dark, scaly outer covering is disappearing. Here's one that's progressively more decayed. And finally, one that people might speculate has been subject to a buildup of decay gases and some kind of perhaps explosion at the bottom of the Green River Lake. So here we see a succession of specimens that show us decay in a fossil specimen. But of course, we can also look at this with considerable profit in the laboratory. We can learn a lot about how preservation occurs by looking at animal specimens in the lab. And here's a series of specimens of the bristleworm Nereus, and apologies to any of you who are in a part of the world where you just had lunch or indeed dinner, because if we watch this decay over a couple of weeks, here you can see the rear of the animal becoming fluid, as indeed the muscle tissue starts to decay away. And here we have one that's even more flattened. And of course, at a somewhat later stage, here we have about 2 weeks of decay. You can see just the skin of the polychaete or bristleworm with a number of the bristles. And then if you let these kinds of experiments go for pretty much as long as you're prepared to in a laboratory situation, what you end up with is this. So at the bottom of the slide, you see the jaw elements of the worm, and at the top, you see the bristles that give these bristleworms their name. And of course, those bristles at the top number in hundreds on a bristleworm. They're attached to the several lobe-shaped appendages, whereas the jaws are just a single pair. But the point is here that as the organism decays, information is lost, and we have to stop this process at a relatively early stage in order to retain the kind of information we need to provide evidence of the soft parts, to give us, if you like, an exceptionally preserved fossil. So people have looked at this in a variety of contexts, and a celebrated series of experiments back in the 1970s by Tom Schopf, carried out at Cold Spring Harbor on the Washington State coast, gives us a series of figures of the percentages of different kinds of organisms that might survive. And in the end, as you can see, about 60% of animals in the mud fauna and in the sand fauna and in the rock fauna don't survive, or conversely, only about 40% of animals in these different settings are normally preserved in the fossil record. I should say these experiments were carried out at Friday Harbor, not Cold Spring Harbor. So let's turn to our deposit of interest today, which is essentially the Burgess Shale. So this very nice illustration shows a number of specimens on a slab. We can see a series of worms, the animal Otoya, which is this silvery looking structure. I'm going to see if I can get us a little bit of an arrow here. So we're essentially looking at these things here. This is a Toya, and this animal here is a soft-bodied animal called Eldonia. And these organisms are the kinds of things that would decay very rapidly under normal circumstances. So here we are at the Burgess Shale. So on the left-hand side, Charles Walcott, who discovered this famous locality in the Canadian Rockies in 1909 at the end of a field season, and went back there in subsequent years and collected a very large number of specimens which reside in the Smithsonian Institution in Washington. And you can see me in the same hallowed ground about 100 years later with Mount Wapta in the background, and I'm standing on the now Talus slope directly in front of the Walcott Quarry. So this is the general appearance of the area, and if we pick out the Walcott Quarry, it's here. It's at the other end of this arrow. You can see I'm not so good at this. There we are. And if we scan across, we can see a close-up of the quarry just here, which, I'm sorry, right there, which is picked out by a snow band. And again, panning across the general area, we can go from the Walcott Quarry right across to the front face of Mount Field. And indeed, I've had the privilege of exploring this area and working down the front face of this mountain to localities across the slope of Mount Field. And some of these are out here in the areas around the end of the quarry. So Walcott's quarry is out here, and we walk across the front face of the mountain and down the slope onto the front face of Mount Field. So this is by way of saying that Walcott thought his original quarry was a unique deposit, but in fact exploration in the area by Royal Ontario Museum expeditions has established that there are many more localities in the general vicinity that also yield these fossils. So this is what the front face of the quarry looks like. The small silver dot in the middle is the size of a US quarter. It's presumably a Canadian quarter. And you can see the bedding planes, which represent a series of depositional events. And what's striking about these, apart from the fact that they're events as such, so they represent single large influxes or smaller influxes of sediment, the other thing that is particularly obvious is that there's little or no disruption of that sediment. There's no burrowing or bioturbation. So once the carcasses get buried, they've a pretty good chance of avoiding being scavenged and removed, which is essential to this kind of exceptional preservation. So here are a series of diagrams from one of my PhD advisor, Harry Whittington's papers back in the 1980s, which shows the fossils themselves out here at the front of the diagram. Those are the animals gaily getting about their business. And then we'll next see a sudden removal of sediment where it's transported downslope to wherever it's deposited here. And as you can see, the animals are oriented in a variety of different orientations to bedding when they end up in the sediment that is in the Walcott Quarry. And that means you get views of these specimens from different orientations, from the side, from the top, from head on, which of course is very useful when we come to think about how these things are preserved in the rock. So let's have a look at a few fossils. The specimen here on the left-hand side is Morella, the little lace crab as it's called. And this one here is Canidaspis, which is a slightly larger, large shrimp-sized bivalved arthropod. So if we focus for a little while on Canidaspis, one of the advantages of the type of transport and burial is that sediment gets into all the different parts of the specimen. So the front of this animal is to the right-hand side, and the trunk and tail are at the rear. And if you remove the outer carapace, the valve, you can see a series of appendages, little flap-like structures underneath. So you can do a kind of paleodissection of these specimens. You can remove information and reveal other features in the rock. And that's extremely valuable when you come to try and work out what's going on. So the other thing we can do is to take advantage of the fact that the rock splits and leaves part of the fossil on one side and part on the other. So here, if you like, is the part and counterpart, two sides of the same specimen. And it's possible to prepare these. In this case, I removed these first three appendages here, which are right-hand appendages, and exposed a complete suite of left-hand appendages as illustrated in this bottom figure 3. And likewise, on the right-hand side, I removed all these appendages to reveal the right-hand appendages underneath on this side, which you can see in figure 4, bottom right. So this is almost like putting your shrimp or lobster-like animal on a table and cutting it literally vertically down the middle and separating the two sides. So you can see how you get substantial additional useful information in this way. As I said, a kind of dissection of the specimen. So we can illustrate the importance of the exceptional preservation in various ways. Here's one. This is a cartoon showing a representation of the variety of different kind of animals we find in the Burgess Shale. And if we take away everything here that we would classify as soft-bodied, everything that has no hard skeleton and therefore is not preserved in the normal fossil record, this is essentially what you're left with. So these are the animals that have a hard mineralized skeleton and which make up the normal, if you like, Cambrian fossil record, the hard parts only. And as I've indicated here, Simon Conway Morris did a very elegant survey, a sort of census in 1986, where he showed that as many as 86% of the Burgess Shale genera, and perhaps even as many as 98% of individuals, because many of the soft-bodied animals are by far the most common, would not normally be preserved. So there's a huge advantage in having the complete soft-bodied community as opposed to having what we get out of the regular Cambrian fossil record. So the animals are preserved for the most part as films of organic material, what we might call a kind of carbonaceous residue, a residue made of carbon compounds. It reflects the original composition, but it's altered over time by various processes of organic diagenesis. Associated with that organic material tends to be films of clay minerals, and these are picked out in different ways. But here, for example, you can see the lace crab morella. The specimen is over here on the left in the top left-hand corner. And in this case, what we're doing is showing an elemental map, a map of the distribution of silicon. And as you can see, it very nicely picks out the outline of the original specimen seen here. And that outline is picked out pretty well by a number of different elements. And these show clearly that clay minerals are associated with the surface of the organic material. So clay minerals appear to play a role in Burgess Shale preservation. Research is ongoing on precisely what that role might be, but we've done experiments in the lab showing how clays can inhibit the growth of bacteria. So here we have low growth to high growth on the left-hand vertical axis, and we've put in calcite and clay and a control. So here's our calcite, here's our clay, and on the right-hand side, a control. And in this little key here, we can see concentrations. So if we look, for example, at the highest concentration, which is this purple block, it's quite clear that there's much lower bacterial growth where you have clay present in the experiment. And if you home in on particular clays, as indeed we did on the right-hand side here, using calcite and indeed a control without clay or any mineral, you can see that berthurine, which is a transformation of kaolinite here, these two clays tend to play a significant role in dampening the growth of bacteria. And therefore we conclude, and we've lots of other evidence of this sort of process, we conclude that the clays inhibit decay in some way and may bond themselves to the organic material in the fossil. So clays are important. This diagram shows numbers of exceptional preservation. So you can see up here, marine Lagerstätten. Lagerstätte is a German word for an exceptional fossil deposit. Down here we have Ediacaran and Cambrian, and suffice to say, the graphs are highest here in these intervals of time than they are subsequently. And this simply shows that exceptional preservation is more common in the Cambrian and indeed the late Proterozoic in the Ediacaran than it is subsequently in the fossil record. And here, this simply shows a relatively small number of exceptional preservations. People have worked this out with very large samples and shown that it holds true. So the Cambrian is unusual, not just for its animals, but also because it tends to favor exceptional preservation. So that's borne out by the large number of different exceptional preservations in the Cambrian geological record. This is a map of how the world looked during the Cambrian. The continents, of course, were in different places and had different outlines. We can see the Burgess Shale here, a very famous deposit, and several indeed of them in China, including Chengjiang. These deposits are a little older, but you get the impression that there are a large number of these exceptional preservations during the Cambrian, that they're scattered across the globe, but that many of them, as you can see, fall within relatively low latitudes, within the tropics. And there's good evidence that this was the site of the degradation of continental masses and bringing a lot of clays into the oceans. And this may have been a factor in explaining why there are so many of these exceptional preservations in Cambrian rocks. Lucky for us, because the Cambrian explosion is arguably one of the most significant events in the history of life, and it just turns out that it happens to be very well evidenced in the fossil record. So here are a few of those Cambrian localities, just to emphasize the point. Here's the Burgess Shale here. Here's the slightly older Chengjiang formation in China. And we'll come later on to fossils from a relatively recently discovered deposit in Morocco called the Fezouata formations, which is Ordovician in age as opposed to Cambrian, and shows that some of these phenomena persist into later rocks where the conditions happen to be right. So what about these other preservations? Here's a trilobite-like animal, or a trilobite called Noroya, with just 2 parts to its carapace. This is a Burgess Shale specimen. On the right-hand side, you see a specimen from Chengjiang, where new discoveries are pulling out specimens that are preserved not only like the Burgess Shale material as an organic carbonaceous film, but also replicated in pyrite. And pyrite allows us to view these things in 3 dimensions and rotate them on a computer screen, which gives powerful new information. And indeed, China and Canada have been the source of new material. This is material here from Marble Canyon, close to where the original Burgess Shale Walcott Quarry lies. And this represents examples from the Xinjiang formation, which is a new discovery in China of Burgess Shale type fossils, one of many examples of these remarkable fossils in that part of the world. So in a sense, we're spoiled for new information, but I'm going to focus on the Burgess Shale because in a sense, it's the classic. And in some ways, of course, I have a history of working on that particular deposit. New things inevitably are coming out of these new discoveries, but new things are also coming out of some of the original deposits. So this is a giant arrowworm, which I described with Jean-Bernard Caron of the Royal Ontario Museum just a few years ago. And this is a specimen which is represented in the Burgess Shale, but also in localities that we discovered back in the 1980s, as opposed to new localities that have been found since. So new animals continue to emerge. This thing has a forbidding array of grasping spines, which it used to attack its and grasp its food. And indeed, arrowworms are ubiquitous in the modern oceans, although at much smaller sizes. So historically, what's the Burgess Shale shown us about the early evolution and history of life on the planet? Well, here's Harry Whittington, bottom left, my advisor in Cambridge. And this is one of his iconic fossils, this animal Opabinia. And I still remember seeing him present this at a conference in the 1970s when the entire audience simply burst out into laughter. And this is evidence, if you like, it witnesses the fact that back then, not so long ago, these Burgess Shale fossils were considered to be extraordinary and really very weird. And indeed, that's echoed in some of the narrative in Stephen Jay Gould's book, A Wonderful Life. But now, of course, we don't really consider them terribly weird because we've become used to seeing extraordinary things coming out of the Cambrian. But also, perhaps more importantly, we have a rather better understanding of their significance and indeed what they mean. So here are some of the originally described weird wonders, which are highlighted in Steve Gould's book. But now we tend to recognize things as representatives of modern groups, or at least early offshoots of the lines leading to modern groups, what we might call stem taxa, taxa which come, or animals which come off the stem. So we think now of Hallucigenia, for example, as a lobopod, and I'll come back to that a little momentarily. And the same story is told by the arthropods, the most abundant group in the Burgess Shale, and indeed the most abundant group around today, because even the arthropods look somewhat unfamiliar to our modern eyes because they're unlike arthropods living in the oceans today, but nonetheless, they belong on the stem leading to modern arthropods. They're offshoots that subsequently became extinct. So back in the day, in the late 1970s, the Burgess Shale arthropods were illustrated like this, and you can see that Whittington drew a series of individual lines crossing the boundary between the Precambrian at the bottom and the Cambrian. This would be the Cambrian explosion, and his argument here was, we've no idea how these things are related. We can't put them together. So instead of making a kind of branching diagram, what we call a phylogenetic tree, this was caricatured as a sort of phylogenetic lawn. It just emphasizes how we wrestled with this problem of these animals looking so different to anything that was around today. If you can't make sense of them, you argue that they're some kind of independent lineage or line. And that led Stephen Gould to think about a model of the evolution of Cambrian animals, which got away from this traditional idea of a cone from simple origin branching toward the present day to something showing a huge array of form, morphology or form being on the horizontal axis here, and that most of these things became extinct, leaving just a few lineages which gave rise to the groups that we see today. And we can see that diagrammatically illustrated here. So we might consider this as the traditional view, this as Gould's view with a huge array of morphology in the Cambrian, and this really is our present day understanding, where there is indeed an explosion, but the range of form doesn't really exceed much beyond, if indeed exceed, what we have at the present time. So this is a way of saying that most of the variety available to groups, the different kinds of morphologies we might find, were established in the Cambrian, and that evolution was constrained by those boundaries through time to the present day. So here's our Burgess Shale. Let's explore some of these, what were originally described as weird wonders, to illustrate some of these ideas. So this is Aishea, which we now know is some kind of lobe-like organism, and indeed Whittington identified it as such. This animal here, Microdictyon, was originally described from little mineralized plates. You can see one here. And when first discovered, people had very little idea of what these might have belonged to. Were they parts of a single organism? Were they some sort of support structure for a colony? And of course, with discovery of these remarkable fossils in Chengjiang in China, specimens were found with these weird plates on either side of a worm-like animal. And here you see it, still of course called Microdictyon. Here are these paired structures on the divisions of the body. And this brings us to one of the animals that really struck Gould as being strange and peculiar, the one that Simon Conway Morris called hallucigenia, and which was originally reconstructed with the then available evidence as an animal standing on stilts on the Burgess Shale seafloor. Subsequently, new specimens showed that the structures here, which look like individual tentacles, are actually paired, and you can see them at different places along the structure here. And this allowed a new reconstruction of hallucigenia, which turned it the other way up and demonstrated that, weird though it might look, it was actually also some kind of lobe-shaped organism, but in this case with spines for protection, as opposed to the kind of disc-like structures that we see here in Microdictyon. So these things are obviously related, and indeed there are very nice reconstructions of hallucigenia now with new insights based on new specimens, which of course is the way to go forward. Here's Microdictyon, here's Onocodictyon, and here's a modern lobopod, a velvet worm or onychophoran. These animals live on land, not in the oceans, in forests in the southern hemisphere, and there's clear evidence now that these are also part of the same group to which what were originally thought of as weird wonders, totally unexplainable organisms. It just turns out that they happen to be part of a modern group, in this case one that lives on land as opposed to in the oceans. Here's a second example. This is Anomalocaris, strange crab, as the term originally coined indicates. What you see here was originally interpreted as the body of a shrimp-like organism, and this thing is 10 or more centimeters long. So we can see in a reconstruction by Charles Knight in National Geographic in the 1940s, here is the Anomalocaris. Let me just, try that again. Here it is here, and it's attached to the structure, which is the valve or bivalve carapace of what we now know as a different animal. So this tail was indeed interpreted as the body of some kind of shrimp. Then there were jaws flying around, if you like. This is now assigned to Herdia. I should say that we originally had a taxon Anomalocaris. There are now about 25 different ones from different parts of the world. So since our work in the 1980s, many of these animals have been discovered in different localities around the world. And this jaw-like structure was originally reconstructed, as you can see here, as some kind of jellyfish, or it looks almost like a pineapple ring floating around in the Cambrian oceans. So here you see our pineapple ring or jellyfish, but of course, what's obvious when you look at it is that there are teeth in the middle of it. So it's clearly some kind of strengthened jaw-like structure. And Whittington and I then discovered specimens which showed the jaw-like structure in association with appendages, which are not so easily seen in this specimen, and managed to reconstruct the animal rather like this in a reconstruction from David Norman's 1994 book. So this is Anomalocaris, or at least an anomalocaridid, as we now understand it, a large, largely soft-bodied animal with a toughened jaw and toughened raptorial appendages at the front end. So these things are mighty predators, and indeed you've got to think of them as animals that got to a meter or larger in size. These were at the top of the Cambrian food chain, and there's evidence that they occasionally bit trilobites. Here's a trilobite with a W-shaped bite out of the rear of it, which is essentially here. Not easy to see in this slide. And I had the delight of working with Whittington and a Japanese television company years ago to explore how models might reproduce these bites. And we worked on trilobites, which the company kindly created, and we acquired them for our experiments. Here's our model, or their model animal with the jaw, and here's a bite out of a real trilobite, just showing you what Anomalocaris or Anomalocaridids were capable of. I don't think these things preyed on trilobites normally. I think they were more intelligent than that, because trilobites, of course, have this nice hard skeleton to protect them. I think what essentially happened was they preyed on softer organisms for the most part. And here's a model of Anomalocaris swimming, which was again created for this television program and gave us some very useful insights into its biomechanics. These animals, as I said, occur all over the world. This is one which was kindly named after me, Anomalocaris briggsii, which comes from the Emu Bay Shale in Australia. Here's an example from the Chengjiang deposit in China, nicely reconstructed here. And here's another anomalocaridid known as Herdia, which comes from the Burgess Shale and has this extraordinary carapace, which you see here, made up of a series of elements which extended out in front of the animal itself. And this is based largely on the work of Alison Daly, who's now the international expert on animals of this kind. So let's move then from Burgess Shale localities in the Cambrian to these new localities that were discovered by a Moroccan geologist and collector, and my former postdoc, Peter Van Rooij, who's now based in Belgium. And here we are in the field, Peter and Jacob Winter and myself in Morocco. And of course, the beauty of working out here is that you have very nice exposures with very little vegetation, and you can follow a bed across the outcrop, as you can see here, with three of us standing simply to indicate the continuation of a bed along strike, as geologists would call it. And these layers tended to yield anomalocaridids. And the neat thing about these anomalocaridids in Morocco is that they occur compacted in shales on the one hand, but they also occur in giant concretions. And this is one here in the lab here at Yale. The concretion itself is about 1 meter long. And Peter Van Rooij's very careful preparation over many weeks of this specimen revealed its features, including 2 pairs of flaps along the sides and these filamentous-like structures down the dorsal surface, cetal blades in this extraordinary animal. Here are the grasping appendages. So you can see the main stem of it is here, and here are the blades which extend down, and then here, indeed, filaments extending out, and you can see little filaments on the big filaments. So this is a kind of fractal-like structure on the appendage. And what this made very clear was that this extraordinary animal, which we called Ediacaran, is obviously some kind of anomalocaridid, or radiodont, as they're now called, based on their circular jaws, but that unlike the others, which appear to be predators, this animal has an anterior grasping appendage, which is modified with all these filaments or teeth as a filtering apparatus. So this may have been one of the earliest large organisms to be essentially filter feeding in the oceans. It's like a giant Ordovician basking shark or whale that's collecting food on the filaments on its limbs. Other Burgess Shale type things also occur in the Fezouata formations in Morocco, including this little arthropod called Furca, which is very similar to Morella and indeed clearly closely related. And remarkable mollusks like this animal Calva pilosa, which I described with Jacob Winter and Peter van Rooij, and indeed our colleague in Oxford as well. And you can see that we have a valve at the front end, and we have a series of spiny spicules down the length of the trunk of this thing. And here's the traces of the jaw, the sort of rasping structure or radula, which is down the length of the animal itself. And this is a reconstruction of this thing prepared under Jacob Winter's guidance. And these things, again, represent very early offshoots of animals that are heading toward modern worm-like mollusks, what we call aplacophorans. So we're discovering new organisms, we're discovering new localities all around the world. We have a much clearer picture of the Cambrian explosion. We're also rediscovering old localities. These are trilobites from Northern Vermont, from one of the first localities, indeed the first locality to ever yield Burgess Shale type fossils in the 1850s to 1880s. And this locality was rediscovered recently by Giovanni Pari, my colleague, where you see us here. And this splits beautifully into enormous slabs, which you can see here are basically, human size. The fossils are extremely rare, but we're discovering here new things that were unknown from this locality before, including some kind of radiodont or anomalocarid, and indeed worm-like animals like the preopulid atoya and little bivalve arthropods. This animal we're probably going to call Vermontcaris, and it looks somewhat like this, although this is not Vermontcaris itself. It's a different arthropod, just to give you a sense of what these particular animals look like. So, what caused the Cambrian explosion? This is still, in a sense, an unknown, but Eric Sperling has drawn analogies between what is required to essentially fuel fires, a mixture of oxygen and fuel and heat. So in terms of
Sorry, everyone. I think we've got a connection issue with Derrick there. Hopefully he will be back in a moment to finish off what is the last few slides of his talk. In the meantime, just a reminder, do put your questions, if you have them, on the chat. We've got a few coming in already, and click on the right-hand side. You can see the little question button there. Hopefully Derrick's connection will be sorted in a moment. In the meantime, just a reminder that our next talk, which I'm about to make pop up in the chat, so there's a link that you can click on, is by Professor Gabriela Mangano from the University of Saskatchewan in Canada. And her talk is going to be titled An Alternative Reading of the History of Life, a Perspective Through the Lens of the Trace Fossil Record. Those trace fossils that Derrick was discussing because of their absence in the Burgess Shale. trace fossils would be churning the sediment, and they're not seen there, which is one of the reasons we see such lovely fossils, which he's so kindly guided us through this evening. I think he's shortly going to be reconnecting. We're on his final few slides, and then we will get to some of your questions, but do keep them coming in. I will also say at this point that if you've enjoyed this evening's lecture, or indeed this morning's or this afternoon's lecture, depending where you are in the world, and you are able to, please do consider donating to the museum. I'm going to make a little link pop up now in the chat. The Oxford University Museum of Natural History is a charity, and as well as putting on public engagement events like this one, we also, of course, have to care for our collections and also the very lovely building that we're in. And at that we can see Derrick's back in the room, not a problem. And so I'm going to make myself disappear so we can hear more about Eric Sperling and his colleagues' theories on the Cambrian explosion.
So briefly, and I apologize, my internet simply crashed. I have no idea why. It doesn't often happen. I was almost done. So evolutionary development, on the one hand, is something that plays a role, changes in the genome, changes in the way that body plans can evolve. Ecology is clearly important because we're concerned here with, I'm trying to get my pointer back. Let me just, bear with me 1 second, if you will. Ecology, because organisms affect the environment. So when predators get going or burrowers get going, they modify the substrate, they change the community, and the environment is changed by the organisms, and that performs a kind of chain reaction where the two play off each other. And then environment itself, it depends on climate and indeed temperature in the oceans. It depends on oxygen. There's no doubt that oxygen was some kind of control on the Cambrian explosion. Certain levels of oxygen are required by certain kinds of organisms. And ironically, food may have been a limiting factor initially because in the absence of particular levels of oxygen, food may also be at a low level and therefore in some sense inhibit that kind of radiation. So the game goes on. We enjoy new methods of analysis. We continue to make remarkable new discoveries around the world, and people are continuing to find new information on the late Precambrian Cambrian environment with fieldwork and with geochemistry, and the picture is gradually becoming clearer, but there's no doubt there's plenty going on that will keep scientists around the world busy for many years to come. So thank you for your attention.
Thank you so much, Derrick, for what has been an amazing lecture, looking at the Cambrian explosion, lovely look at the Burgess Shale, and then some of those newer sites that have been discovered in more recent years. Thank you for that. Do remember to keep bringing your questions into the chat. We'll start off with a nice light question from one of my colleagues in the museum, Imran Rahman, who asks, what is the name of the trilobite above your head that we can all see, lovely and resplendent? All.
It's a trilobite called Terataspis, if I recall correctly, which is from Texas. And it used to be the largest known trilobite, which of course is appropriate for Texas. I think it's now superseded by one from northern Canada, a big isotelid. And this is not life size, it's 3 or 4 times life size. It was given to me by a Peabody model maker who actually rejected it because he thought it looked rather dull. But actually the colors, I think, are perfect because they're more likely to be the sort of camouflage colors a trilobite might have adopted on a seabed.
Well, it looks wonderful from all the way over here in Oxford. Just while some final questions come in, I am just going to do a final reminder of our 2 upcoming talks. The first of which, as I've mentioned a number of times now, September the 9th by Professor Gabriela Mangano, and then wrapping up the first animals series on the September the 23rd will be Edinburgh's Professor Rachel Wood. So 2 brilliant talks there to look forward to. And in the next few weeks, we'll hopefully be making an announcement of what comes next. So keep your Wednesday evenings free. And as always, we love to get a bit of feedback from you. So we'd be interested to hear in the chat. Has this lecture changed your opinion on the evolutionary origin of animals or added to your understanding of the topic? If so, how? And at that point, I will jump to some questions because you inspired many great questions on all sorts of topics. And we will start with clays. Phil from Yorkshire would like to know, do clays play the same role in preservation in the Chengjiang biota as they do in the Burgess Shale?
We think they do. We have a sense that clays are probably important to all of these Cambrian deposits that are preserved in the same sort of Burgess Shale type preservation, i.e. primarily as a carbonaceous organic remnant of the original animal. And
Robin asks, how might the clays mediate the decomposition? Are they controlling in some way what the bacteria can do?
I think, well, our experiments tend to indicate that certain clays, and indeed we have ongoing research which tends to suggest that the clays that cause or inhibit bacterial decay in experiments are the ones that are also most prevalent in the deposits. And of course, we've done that with controls using clays that are not so ubiquitous in the deposits. So that's helpful. We have ongoing work, and indeed there are others working on this kind of problem as well, that would indicate that the clays potentially inhibit bacteria, but also may bind in some way to the organics on the surface of the specimen as it decays, and in that way also perhaps physically prevent access by the bacteria in quite as readily a fashion as would be normal. I should say that critical to decay are not only the bacteria in the ocean itself, but also the bacteria within the specimen. So that's not quite so straightforward, because getting the clays into the animal is a different sort of problem.
And we've got obviously people in the audience who are interested in all sorts of different types of fossils. And Dolores asks, are clays important in the preservation of plant fossils?
Yes. And in fact, very early work by Bob Spicer at the University and others demonstrated that well-preserved leaves very often have a superficial film of clay on them. And if I recall correctly, that may well have also been explored in experiments.
And very glowing support for your lecture, Derrick, in the chat. A number of former students, thank you for tonight's lecture. And at that point, we'll move on to the controversial topics around the Cambrian explosion. Is it real? When did it happen? Why? And all of that. Kaylee Mills starts us off with, are there any theories as to why there are so many examples of exceptional preservation in the Ediacaran and the Cambrian, and what environmental factors might have led to it?
Well, we've dealt in a way with the Cambrian, because there we would argue that it's largely about the effect of clays and rapid burial and a set of circumstances that may be unique to the Cambrian. Then, of course, the question is why in the Ordovician in Morocco? And I think the answer there is, there you have a very substantial thickness of mud rocks, which are similar in a way to many of the settings that yield exceptionally preserved Cambrian fossils, but which are not that common in the Ordovician. So that may be a function of the availability of the right kind of context. So I think we have a reasonable idea of what's going on in the Cambrian. The Ediacaran is rather different, and there are a different set of explanations, and indeed the jury is very much still out. But my colleague Lydia Tarhan has come up with a very interesting idea suggesting that a higher concentration of silica in the Ediacaran oceans may have led to rapid precipitation, because many of the Ediacaran fossils don't yield the remains of the animal. There's nothing organic there. That's not always the case, but it's often the case. And where it's not the case, you're dealing with the rapid preservation of the shape, if you like, which requires precipitation of minerals in the sediment. So these kinds of explanations are coming out of people looking at sequences of rocks and analyzing their geochemistry. I think that's where some of the more novel ideas about why preservation is as it is in the Ediacaran and Cambrian are going to come from.
And a question about Cambrian fossils from Owen, who asks, why do you think so many of the Cambrian fossils, especially those we've been looking at this evening, are segmented, free-swimming, and not burrowers?
Many of them are arthropods. I mean, that's partly a function of the sample, but and it may partly be because burrowing was an evolutionary habit that evolved over time and took considerable time to become fully diverse, so that there may have been fewer burrowers at the time. The other thing that's going on, of course, is that we're seeing an environment where the animals were preserved as opposed to where they were living for the most part. So in essence, the sediment where they're buried, as I showed, is not disturbed because there's low oxygen down there. It's not the kind of environment that burrows like, because the sediment is, in a sense, hostile to life. So there are various things going on. The evolution of burrowers went on over a period of time. Again, my colleague Lydia Tarhan and indeed Louis Boutois and Gabriella Mangano, who we're going to hear from in a week or two, have worked very hard and intensively on the evolution of burrowers. So I'm sure we'll get some good insights on that in that subsequent lecture.
And a question from Sarah, who asks, from someone who hasn't studied these issues, was the Cambrian explosion followed by a mass extinction?
Well, there were some extinctions during the Cambrian. The big extinctions are not Cambrian in age. If you like, the next big one tends to be late Ordovician, but there are constant extinctions. But the diversity curve tends to continue to rise from Cambrian through Ordovician. And indeed, a major preoccupation of many of us is whether indeed there is a sort of hard and fast distinction between the Cambrian explosion and the next big event, as it's been styled, which is it's an awful mouthful, it's the Great Ordovician Biodiversification Event. And that tends to produce a lot of the next rank down from the big body plans to, divisions within those body plans. So is the Cambrian explosion separate from the Great Ordovician Biodiversification Event? That's an ongoing debate, and it's the subject of a lot of research and indeed data on new localities and diversity patterns through time will resolve it eventually. Now,
your inclusion of your videos, your TV work with those lovely reconstructions has spurred a lot of interest. And for anybody who would like to watch that program, my colleague Imran Rahman has placed a YouTube link in the chat where you can see some of those wonderful animations. And I think it's people are asking to know a little more. And Morrison asks, what do you make of Dr. Hagedorn's research suggesting that Anomalocaris were not predators of hard-bodied organisms?
Well, I think for the most part, he's probably correct in the sense that, my own inference is that why attack a trilobite if you've got a diversity of other arthropods around which are much more easier to process and don't have a hard outer skeleton. So yes, I mean, he's done some interesting work on the strength of the jaw. I suspect that Alison Daly and others would argue that it's not quite as weak as James Hagedorn thinks, and that the Anomalocarids or radiodonts could certainly deal with pretty tough organic skeletons. And I think they could break trilobites, but I don't think they, for the most part, dined on trilobites.
Final few questions now. Jane Still asks, you talked about the position, the taxonomic affinity of Opabinia, but Jane wants to know where is Opabinia placed now?
Opabinia is down at the base of the arthropod line. So it's an early offshoot, and most people think it came off between lobopods like Achaea and Hallucigenia and radiodonts like Anomalocaris. So it's a kind of, well, intermediate is not the right word, but it branches off between the lobopod branch and the Anomalocarid branch, and then above the Anomalocaridids would be what we would normally identify as arthropods.
And Caroline is interested in a fossil group you haven't mentioned tonight, but she wishes to apply some of the knowledge and theories that you've talked about between hard and soft-bodied preservation. And Caroline asks, are there any ammonites with the soft parts preserved, or are pictures based on other animals?
There are very few ammonites with soft parts preserved. One of my former postdocs, Isabelle Kruta, who's now in Paris, she described the jaw structures within some ammonite shells where they were preserved intact and were revealed by scanning methods. But as you'll understand, ammonites live up in the water column. They're swimming organisms. When they die, I suspect the carcass floats for quite a while. So normally by the time they get to the sediment surface, most of what's of interest has decayed. And I think that's the essential explanation as to why soft parts are so rare. People get excited about the jaws. They're tougher, of course, and more likely to survive. But the trick is finding them in roughly life position so that you can reconstruct what was going on.
And our final question goes to Emma in what has become a recurrent final question to our many esteemed speakers. And it is a simple one, but sometimes very difficult, because there are paleontological colleagues in the audience who might be perturbed by your answer. And the question is, what is your favorite type of fossil?
Well, I suppose, I've worked on, an awful lot of different kinds of fossils. I would have to say Anomalocaris because, for a number of reasons. One, I had the privilege of putting the animal together with Harry Whittington. Before that, it was bits and nobody had any idea what the whole thing looked like. So that's exciting, and I feel some kind of ownership. And the other remarkable thing about it is that through the work of Alison Daly and others, and through many different discoveries around the world since, it's now turned out to be a really rather diverse group of STEM arthropods, let's say, which are clearly very important in Cambrian ecology and Cambrian communities in a way that, Whittington and I simply had no idea of when we originally started working on this stuff back in the 1980s.
Thank you so much, Derrick, for not only a wonderful, wonderful presentation, but also answering all of those questions. So thank you so much for joining us once more and supporting the museum. Just a reminder to everyone at home that we are back again in 2 weeks' time for the talk by Professor Gabriella Mangano. We very much hope you can make it. Thank you to the hundreds of you who have joined us from all around the world. We hope you've had a lovely morning, afternoon, or evening, whatever it might be. And we very much look forward to seeing you again soon. So on that note, we will bid you a good night and hopefully see you again soon. Thanks again, Derrick.
Thank you.